Vanakkam. Welcome to this course on classics in neuroscience. In this video and in the next few videos, we will be looking at the biology of the cell and the synapse, that is neuronal connections, connection between a neuron and an excitable cell such as a muscle. So, in this video we will look at the grand revolution in cytology. What is cytology? Cytology refers to the study of structure of cells, essentially referring to the study of the inner structure of cells or organelles, what the cell is composed of, and that. Remember, in our previous discussions we suggested, or it was suggested by histologists, that there will be this grand breakthrough where cell and its and the details of how cells and signaling happens between cells will be discovered later. This was made sometimes in the 1850s, 1860s. Remember that? In histology, this was promised. This was the grand promise that cells and the details will be revealed sooner or later. Right? This was the promise made by the histologists of the 1850s and 1860s. At that time, it would have been considered sheer imagination, absolutely no evidence to support this idea. We mentioned this in the previous discussion, or the previous weeks we have studied this. So, cytology refers to the study of the structure of cells. This was essentially carried out using light microscope. The pictures similar to the one shown here is what is usually obtained with the light microscope. There is a tremendous amount of limitation because light is used to study. So, there are optical limitations that challenge any possible observation of the inside of the cell, or the organelles may not be easy to visualize just with the help of light microscope. Of course, this situation can be improved by techniques such as differential centrifugation that will show different organelles differently. Using this method is called as cell fractionation, and then you can study this. This provided some insights, but still even as late as 1949 or the late 1940s, whether the Golgi body is real or is it an artifact, Golgi body, is it an artifact? It is just an artifact of light microscopy is what was suggested by some people. So, the major limitations seem to be the unavailability, not the availability, but rather the unavailability of appropriate methods to study the inner structure of the cells. This was the major limitation, and until the mid 20th century, until the 1950s, practically light microscope was the only method that was available. In the early 1950s, between 1952 and 56, electron microscope transformed many fields or subfields of biology, especially cell biology. Actually, it transformed many other fields other than biology, but here we are interested in cell biology. So, shown here is an early electron microscope. Electron microscope allowed for huge magnification, for example up to 50,000 times, when compared with light microscope, which allowed magnification of approximately 50. So, between the light microscope and the electron microscope, there is a 1000x amplification, there is a 1000x improvement in visualization in microscopy. So, it was possible to observe deep structures within the cell, or ultra structural observations was possible. So, observation of structures within the cell was possible with the advent of electron microscopy. So, the significance of this period or this set of breakthroughs can be compared with what happened in cell theory in the 1840s and 1850s, a period of tremendous amount of development in the field of cell biology, in understanding or in improving our understanding of cell biology. So, this essentially marked the birth of fine structure, cell fine structure, and the field of cellular biology, and it further inspired investigation at the molecular level. So, 1000x that I have written here is improvement over light microscopy. 1000x is not the magnification, 50 is the magnification of the light microscope, 50,000 is the magnification of the electron microscope. Okay. This allowed observation of the cell organelles using the electron microscope. So, several steps happened through the 1950s that various structures or various organelles within the cells were visualized. So, mitotic spindle was first observed by Mazia and Dan in 1952, the earliest observations of subcellular activity and subcellular organelles. Mitochondria was observed by Palade in 1953 and Joe Strand in 1953. Golgi body was observed by Dalton and Felix in 1954. So, Golgi body is not actually an artifact, but rather a real structure. Both smooth and rough endoplasmic reticulum were observed by Palade and Porter in 1954. Structure of muscle cells or how force is produced within a sarcomere or within one strand of a muscle fiber at the nano level, at the micro nano level. This was described as the so called sliding filament hypothesis that showed movement of filaments, actin and myosin over each other, and the connections formed by myosin over the actin to pull, causing the force development. A detailed discussion is outside the scope of this course, but I will share videos relevant to this in the forum, so you can discuss that. Of course, I already mentioned the endoplasmic reticulum that was observed by Palade and Porter. Cilia with the microtubule array by Fawcett and Porter in 1954. Ribosomes at that time was called nucleoprotein particles by Palade in 1955. Nucleus and nucleolus by Porter in 1955. Lysosomes by Novikov and his colleagues in 1956. Finally, the visualization of plasma membrane by Robertson in 1957. So, between 1952 and 1957, almost all or many of the organelles within an animal cell were visualized using the electron microscope. Again, another representation of the same set of observations. So, mitotic spindle was found by Mazia and Dan, mitochondria by Palade, cilia by Porter or Fawcett and Porter, endoplasmic reticulum by Palade and Porter, Golgi body by Dalton and Felix, muscle structure by Huxley, as in Nader-Genk, Huxley and Hansen, myelin by Guerin and Robertson, nucleus and nucleolus by Porter, nucleoprotein particles are lysosomes by Palade in 1955, lysosomes by Novikov, and plasma membrane by Robertson in 1957. Again, another representation of the same thing. What is shown here in animation here, So, various structures being individually isolated or observed. What is shown here is a 3D representation or a rendering of what was observed. So, with this we come to the end of this video. In this video we discussed about the revolution in cell biology or cytology in the 1950s and how electron microscope enabled the visualization of various organelles or the subcellular structures in the 1950s. Thank you very much for your attention.